Okay, could somebody confirm that you can hear me?
Yes.
I can hear you.
Great. All right, let me share my screen. Nope. All right, should see my screen now. I did look up the lab goes to 4:50 p.m., so I had that wrong. I've updated it now. So today what we're going to be doing is continuing the discussion of Chapter 1 and Chapter 10. And hopefully we'll get close to finishing, but not quite. And then we'll next week start with Chapter 1 and start Chapter 2. Any questions about any of that? Let me remind you, you need to have the online plagiarism quiz due this Saturday by 11:59 p.m. You must achieve 100% on the quiz or you'll get 0 on your 100-point infectious disease project. Also remember Lab 00 is due at 8 a.m. Friday. That's this Friday, January 12th. And that's tomorrow. All right. Any questions about any of that? If not, let's continue. Oh, get out of here. Somebody's got a chat there. I'm going to tell you I don't use the chat feature or the emoticons. If you want to ask a question, you're going to have to ask it out loud because I don't see the chat features when I am sharing my screen.
Hey, Professor Wolf.
Uh-huh.
So it was just me. I messaged you about potentially changing the quiz dates from Wednesday to like a Tuesday or Thursday since we have class in those days. So I put it in the discussion, but I don't know if people are checking it. So I also just put it in the chat if that's okay.
Yeah, that's fine.
Cool. Thank you.
Let's go to the lesson. So chapter 1 on microorganisms, we talked about discussing what is a microbe, what is it, what is a microorganism? Let me try that again. Organisms which are too small to be seen by the naked eye. These are microorganisms or microbes, which include bacteria, archaea, yeast, molds, protozoans, some algae, and then viruses and prions. Now, I've talked about a cell, but I haven't discussed what is a cell. A cell is the basic structural and functional unit of all living organisms. A cell is a highly organized compartment bounded by a thin, flexible membrane that we call the plasma membrane or the cell membrane. Cells contain 3 components, and we're talking about all cells have these 3 components. They contain the cell membrane, of course. They also contain DNA for their genome, and they do contain cytoplasm. All cells have those 3 components. Any question about any of that?
Could you repeat the first one, please?
They all have the cell membrane or the plasma membrane. Those 2 terms are used interchangeably.
Thank you.
Cytoplasm and DNA. All right, when we're looking here in these pictures on Earth, where would we not find microbes? Would we find microbes in the water, on the plants, on the rock? Would we find microbes in the ocean water or in the air above the ocean? Would we find microbes, obviously, in the plants or in the ground, in the soil underneath the plants? Obviously, the air again, on the person or the cat, and in the desert soil. Where would we not find microbes? Anyone want to have a guess? All right, nobody's making any guesses. So you're going to find microbes everywhere in all of these pictures, including the ocean, in the soil. You can find microbes down about a mile into the soil. And you can also find microbes about a mile up in the air. Now, that would differ if you have bedrock, but in the soil, it would go down a mile. And that's true for in the ocean floor too. You can dig in the ocean floor and go down about a mile, and you'll find microbes in the ocean floor, assuming that it's not bedrock. Any question about any of that? You find it in the desert sand and the desert air as well. You find microbes everywhere except for this one place here. This is a volcanic vent. And let me blow that up a little bit. You can see the red in there, that's lava. That would be too hot for microbes to exist. And that's about the only place you do not find microbes on the planet Earth. So microorganisms are almost everywhere on the planet Earth. Where is everywhere? An environment where microorganisms are found is on the land and in the water, in the atmosphere, subsurface, on both the land and the ocean floor, and plants and animals. Essentially, wherever liquid water is present, you will find microorganisms. You will find microorganisms where the water is frozen. However, if you go below a negative 20 degrees Celsius, remember 0 is when water freezes, you will not find any microorganisms that are metabolically active because they will be frozen. They will be present, but they will not be metabolically active. And they may not die. Many microorganisms can survive freezing. Not all can, but many can. And you can find microbes up to around 120 degrees Celsius. Remember microbes, remember that water boils at 100 degrees Celsius, at least at standard atmospheric pressure. The highest recorded microbe that we've found that can survive the highest temperature that was known was 126 degrees Celsius. And that is now, gosh, that might be 20 years old. But they found some microbes growing in an underwater volcanic vent. And at 126 degrees is the temperature that water boils at the deepest part in the Pacific Ocean, the Marianas Trench. You have so much water pressure on top of the water in the Marianas Trench that water does not boil until it's 126 degrees Celsius. Normally, if you remember at standard atmospheric pressure, water boils at 100 degrees Celsius. All right, any question about any of that? How many microorganisms are present? In 1 gram of soil, we have about 1 times 10 to the 8th microbes. And I'm actually only talking about prokaryotic cells, the bacteria, the archaea, and no, I'm not talking about the single-celled eukaryotes. I'm only talking about single-celled prokaryotes. So we're not talking about viruses. If we were to add viruses, the number would probably be about 10 times greater. But 1 gram of soil has about 1 times 10 to the 8th bacteria and archaea in it. It would depend on how much moisture is in the soil and how much organic matter is in the soil. With more moisture and more organic matter, having more microbes, and then less organic matter and less moisture in the soil, having fewer microbes. If we were to take 1 gram of ocean water, that's 1 milliliter, do you think we would have more or less prokaryotes than you would have in 1 gram of soil? Anyone have a guess?
Less.
You're correct. Less. The oceans are one of the deserts on the planet Earth, and so we find less microbes and less life in general in the ocean than we do in, for example, 1 gram of soil. How about 1 gram of human feces, more or less?
More?
Yeah,
More.
generally more. It does depend on how much water is in the human feces, but normally human feces coming directly out of a patient or a person will have more microbes than in 1 gram of soil. Now, on the other hand, if we were to look at feces that has been in the desert and all the water has evaporated from the feces, then you would have generally less than 1 gram of your average soil of 10 to the 8th. But usually human feces straight out of the person will have more. And then how about on 1 human being, more or less?
More?
Yeah, more. It's obviously more than 1 gram. And then entire Earth, obviously more. Here are the numbers here. So 1 gram of soil has about 1 times 10 to the 8th. In the desert soil, you'd have about 1 times 10 to the 6th. But even in the desert soil, we will have prokaryotic cells. And then the ocean water has about 1 times 10 to the 6th prokaryotic cells in 1 gram or 1 milliliter of ocean water. 1 gram of human feces, if the feces are fresh, straight out of the person, you're going to have about 1 times 10 to the 11th bacteria and archaea in the feces. Now, if you were to talk about the desert feces, that would have about 1 times 10 to the 6th, which would be the same as the desert soil. For feces, actually 55% of the wet weight of the feces is actually microbes. 1 human has about 3.8 times 10 to the 13th prokaryotic cells in and on them. The entire Earth, we are close to the human population, close to 8 billion. I guess I should change that. It's closer to 8 billion now. And a little under 8 billion. At least that was my last understanding, but I haven't looked recently. So the number of bacteria and archaea on the planet Earth is estimated to be 1 times 10 to the 30 zeros. meaning 1, followed by 30 zeros after it. This does have a name. It's called 1 nonillion. You probably never even heard of a nonillion before. Any question about any of that? Okay, let's talk a little bit about microorganisms and disease. If a microbe causes a disease, we call it a pathogenic microbe. So microbes that cause disease are called pathogens. Most microorganisms do not cause disease. Only a small percentage of all microbes are pathogenic, or a small percentage cause disease. When talking about disease, it's important to make the distinction between an infectious disease and an inherited disease. What is an example of an infectious disease? Can anyone give me an infectious disease? All right, you guys can think about that. An infectious disease is one that is caused by a microbe, and that's why it can spread from one patient to another. An
What
inherited,
about, is it COVID, like COVID or flu?
yeah, COVID and the flu are both infectious diseases.
AIDS, Hep C.
AIDS and hepatitis C are infectious diseases also. It's curious, you guys are all naming a disease caused by viruses, and that is a microbe. Strep throat would be another infectious disease. I'm just mentioning this because this is caused by a bacteria. An inherited disease is one caused by defective genes. So a child would be born with an inherited disease. They would not get it from a microbe. What is an example of an inherited disease?
Huntington's?
Huntington's disease is an inherited disease. And then
Alpha thalassemia?
yes, another inherited disease that we'll talk about later is sickle cell anemia, where the child, if it's born with sickle cell anemia, it has 2 genes which are defective, causing the child to get sickle cells, his blood cells to become sickle cells. Any question about any of that? All right, here we're looking at some microorganisms. Let me blow this up. Oops, didn't go.
I have a question.
Uh-huh.
Sorry. So would Lyme disease be one inherited, or would that just be because you can get it from, I heard you can get it from being bitten by a tick.
Can you say it again? I didn't hear what disease you're talking about.
Lyme disease?
Lyme disease is caused by a bacteria that you get from a tick bite. So it is an infectious disease. Okay. All right. Here we're looking at different microbes, how small they are. We actually have a scale here. This is bacteria, probably E. coli, which is a little bit larger than your average bacteria. This one is about 1 microliters long. Come on, mouse. That one there is a little longer. It's either 2 to 3 micrometers long, but the width is less. Here is a fungus, and there's actually a cell. Right there's another cell. And there you can see this is larger. That cell there is over 50 micrometers. Actually, that's probably about close to 150 microliters. There is a microbe. That looks to me like a white blood cell, a human white blood cell. And There's its size there. That's about 10. And so that would be 70, 80 micrometers long. Here is a microbe. And you'll notice that this is a colonial microorganism, a microbe that lives in colonies. And this is one parent colony right here. Each of these dots is a cell. And this parent colony has made 6 babies, 1, 2, 3, 4, 5, 6 baby colonies. The parent will open up, the babies will come out, and then they will colonize some other location. And then here is a cell. This looks like a T4 lymphocyte, which is a helper T cell. And then these red, oh, come on, red blobs are HIV virus, which is also a microbe. Any question about any of this? Whoop, that didn't work. Why isn't that working? Okay, microbes play a number of beneficial roles on the planet Earth, as well as beneficial roles for humans. They can decompose organic waste. We mentioned that before. They can be producers in the ecosystem, and they can be 2 different types of producers. They can be photosynthetic producers, engaging in photosynthesis, or they can engage in chemosynthesis. There would be different cells doing each. Microbes are involved in the nitrogen cycle. In fact, the nitrogen cycle would not happen without the microbes moving and converting the nitrogen from one form to another. Microbes can be used to produce substances for man, such as industrial chemicals. 2 examples, ethanol and acetone, can be made by microbes. We can use microbes to make biological insecticides. We can use microbes to make fermented foods. In fact, to get fermented food, you have to have the microbes fermented. Some examples of a fermented food: vinegar, cheese, bread, beer, and then everybody's favorite chocolate. You probably didn't know this, but chocolate is fermented. In fact, brown tea is fermented too. There's a lot of products that are food that is fermented. We can use microbes to produce products used in manufacturing, such as cellulase. We can use microbes to produce antibiotics, such as penicillin G. And then we can use microbes to produce other products used in medicine, like insulin. All human insulin today is made or grown in a microorganism. They took out the human gene for insulin, put it in a microbe, and then produced the insulin in the microbes. That's been going on for a large number of years, because the first time my father, he was a diabetic, took insulin, he got it from insulin that had been obtained from other humans. So it was human insulin. And then after using that for a few years, I'm not sure how long, he became a diabetic sometime in the 1960s. I don't remember exactly when, because obviously I was too young at that point. But they then switched from using human insulin to obtaining lamb insulin, and they just discovered that it was easier getting insulin from a lamb than from a person. So they gave him that insulin, and he used that for a number of years. And then they switched to microbial made insulin. And so that's been going on for a large number of years now. I would say over 20, probably about 40 years, maybe even 50 years now. I don't know when microbial insulin became available, but the point is it is made in microbes. Biotechnology is the use of microbes and organisms to produce food and products for human use. Most people don't know this, but biotechnology is actually centuries old. We've been using biotechnology for a long time. The genetic engineering is a new technique of biotechnology where we put genes into an organism, particularly into microbes, but you can put genes into a person. Applications of genetic engineering include producing a variety of proteins from bacteria and fungi, such as various vaccines, or you're making an enzyme. You can also use microbes to help with gene therapy, where you're replacing someone's defective genes. There are some children who are born with defective immune genes. So their immune system doesn't work properly. Usually, the immune system is working, at least to some degree, but it's not working properly. So it's not working at 100% normal. And you can correct that by putting in the defective genes into the patient. And that we call gene therapy. And then you can use microbes for genetically modified bacteria or genetically modified organism where you put genes in the cell, in the bacteria. And we sometimes do that for protecting crops from insects or protecting crops from freezing. Another example of a genetically modified organism, you probably have heard of it, the flavor saver tomato. It had a gene added to it that kept the tomato from rotting as quickly as it normally does. And it was initially, the supermarkets loved it because the tomato could be kept ripe for a longer period of time on their shelves. However, it must not have been popular with the public because I have not seen the flavor saver tomato in the stores for a long time. Any question about any of that? When talking about microbes, they do have an ecology associated with them. Microbes can recycle carbon, nitrogen, sulfur, phosphorus, and other nutrients that can then be used or reused by plants and animals. And it is the microbes, as well as the fungi and mushrooms, which decompose carbon, nitrogen, sulfur, and phosphorus, and then make it available so that other organisms can use it. So that it is largely the microbes as well as the mushrooms that decompose all living organic matter. And we talked about it in depth about the nitrogen cycle, how microbes convert one form of nitrogen to another in the ecosystem. These are all examples of microbial mediated biochemical cycles that support life on Earth. You would have a hard time having life on Earth if the microbes did not recycle carbon, nitrogen, sulfur, and phosphorus. The nutrients would be probably broken down by something else if the microbes didn't do it. Like you spill glucose in the soil, there will be something that'll eat that glucose up. The microbial-mediated biochemical cycles that are run by microbes were first discovered by Winogradsky and Bajorink in the 1800s, late 1800s, 1880s, and the last one, 1892. They were the first to show that, I think they first showed it with nitrogen and sulfur are moving around in the ecosystem because of microbes. And the reason why they were able to discover this is they were studying microbes in a different manner than most other scientists. Most scientists study one pure species of a microbe, or any species really, and that is they don't study a mixture of them. And the reason for doing that, if you study a mixture and then the test becomes positive, you don't know which species in the test made the test positive. But if you only have one species in the tube and then the test is made positive, you know that one species caused the positive result. Well, Winogradsky and Bajorink were studying microbe communities, meaning large numbers of different species of microbes. And that's why they were able to find the nitrogen and the sulfur cycle, because these cycles happen by many different microbes. Any question about any of that? Microbes can be a benefit to human welfare. One example is bioremediation. Bioremediation literally means life and to remedy. Here we have an oil spill, and what the two guys are doing is they're spraying bacteria on the oil spill for the bacteria to digest the oil and then convert it to carbon dioxide and water. We've talked about how microbes digest the sewage and then convert it also to CO2 and water. Well, you can use bacteria and other microbes to control pollutants like this oil spill. You can also use microbes to control pollutants like mercury, where mercury is not degraded to something other than mercury, but elemental mercury, which is toxic, highly toxic, I should say, can be converted by microbes to an organic form of mercury, which is less toxic. It still is toxic, but it's less toxic. All right, any questions about any of this? You can use microbes to make biological insecticides. These are alternatives to chemical pesticides, and they're usually less toxic than man-made chemical pesticides. You can use them for preventing insect damage to crop, as well as preventing disease transmission among crops. One example is Bacillus thuringiensis, which is actually in this package. There it's shown there. Bacillus thuringiensis causes an infection in insects, which is fatal to many insects. However, Bacillus thuringiensis is harmless to humans and other animals and to plants. My father actually used Bacillus thuringiensis once I think it was in the 1960s, but don't ask me when. I don't remember. I was too young. Tells you I was young in the 1960s, but what he did was he used Bacillus thuringiensis to put it on the apple tree to prevent the apples from getting worms. The trouble was he was too lazy and he only put it on the apple trees one time. Even if you're using a chemical pesticide, you have to spray the apples every 2 weeks or else you'll get worms. So putting it on the apple tree did not totally work. All it did was reduce the number of worms. And another trouble with Bacillus thuringiensis, besides it has to be applied about every 2 weeks, is you have to apply it every time it rains. And he was growing his apples in Western Oregon, which is like Western Washington, meaning it rains a fair amount of time. And what happens is the rain washes the Bacillus thuringiensis off the apples. And so after the rain, you need to reapply it. Any question about any of that? All right, if there's no questions, I'm going to change gear a little bit and talk about the naming and classifying of microorganisms. The naming of all organisms began by this gentleman, and he also was the first person to classify all organisms into different classification schemes. What he did was he classified all living things into either a plant or animal, and then he classified plants and animals into smaller groups, like there would be mammals and primates. Then there'd be a genus and then a species. Does anybody know who this is? Anybody? I'm sure you had that in biology. What is it, 160? All right. This is Carolus Linnaeus, the first person to begin the classification system and the first person to begin the scientific naming system or the scientific nomenclature. He liked Latin so much that he Latinized his name. His real name, I think, was Carl Linné. I think he was Swedish, and he Latinized it to Carolus Linnaeus. Surprisingly, your textbook calls him Carl Linnaeus, meaning they Latinized his second name, but his first name they kept unlatinized, which I'm not sure why they did it that way, but that's acceptable. So his system for beginning the scientific name or nomenclature was so good, it's still in use today. However, his classifying organisms into different groups is no longer used. We no longer classify all living things into plants or animals. We quickly broke that down into, well, quickly, but they broke it down into 3 groups from 2, plants, animals, and microbes. Then they broke it down into 4. I'm not sure what the 4th was. And then they broke it down into 5, plants, animals, fungi, plants, animals, fungi, protists, and bacteria. That would be the 5, or Monera for bacteria and archaea. That grouping then went into 6, where we have bacteria, archaea or archaea bacteria, and then plants, animals, fungi, and protists. And after that, they went to the 15 kingdom was produced, not produced, but suggested. Then there was the 20 kingdom system, and then 30 kingdom system and higher order systems. So the kingdom system is in disagreement for how many kingdoms there are. And it depends which author you use. And scientists have been in disagreement for how many kingdoms there are for at least 20 years. It might be going on 30 years now. Any question about any of that? All right. So Carolus Linnaeus, when he named all species, he gave them two names. The first would be its genus name, and the second would be its specific epithet. The species, like Homo sapiens for man, is two names, the first being the genus, the second name being the specific epithet. You can't say sapiens is the species name because sapiens is not the species name. The species name is Homo sapien. Any question about that? All right. So this is called the scientific nomenclature or the binomial nomenclature because each species has two names. Binomial means bi means two, nomio means names. Whenever you're using the scientific name, the name should be italicized or underlined. So if ever you're writing it and you can't write italics, you underline it. The genus name of a scientific name is always capitalized, and the specific epithet is always lowercase. All scientific names are Latinized, and they're used worldwide, meaning whatever country you're in, whatever language they use, they will use the same name for the scientific name. So humans are always called Homo sapiens when we're using the scientific name, regardless of what language you speak. You would say Homo sapien for a human. The name of a species can be descriptive or it can honor a scientist. For example, Homo sapiens is descriptive. Homo means a man-like thing. What's the term I'm trying to think of? That walks on two legs. Sapiens means wise. So homo means man that walks on two legs. Sorry, that's what I meant to say. Sapiens means wise man. Or wise, yeah, wise man. Escherichia coli is both. Coli is descriptive, and you can find E. coli growing in the gut, and coli stands for colon. Escherichia is honoring a scientist, Dr. Escher, or Escherichia, Escher. I don't know how you say his name. He was an early microbiologist. Staphylococcus aureus is descriptive. Aureus means golden, and Staphylococcus aureus is gold-colored cells. And then it's coccus, that's descriptive too. That means the cells are in the shape of a round ball. And Staphylococcus means that the cells are round cells, but they're in a grape-like cluster. Any question about any of that? When you're using the scientific name or the binomial nomenclature, the first time you use it, all scientific names should be spelled out. However, the second time you use it, you may abbreviate it with the first letter of the genus, and then you write out the specific epithet. For example, Staphylococcus aureus and Escherichia coli are found in the human body. And the second time you use Staphylococcus aureus, you can abbreviate it S period aureus. And Escherichia coli is abbreviated E. coli. I should mention that E. coli is a little bit of an exception, and it's so well known that even scientists, even the first time they use it, will sometimes write E. coli. Because to tell you the truth, I think more people know it by that name than know it by Escherichia coli. All right, so let's move on to classifying microorganisms. Before we talk about classifying microorganisms, we need to classify or take a look at the classification of all living organisms on the planet Earth. All living organisms can be classified in the three-domain system. Three-domain system is also called the tree of life because the domains branch like a three-trunked tree with different branches coming off the trunk. The three domains, or the tree of life, are the domain bacteria, the domain archaea, and the domain eukarya. Each of these domains is classified by the sequence of their DNA. So the domain is defined, and we can classify all species into their domain just by looking at the sequence of the species domain. All organisms within a domain will have a more similar sequence of DNA to all other members of that domain than to any species in another domain. Is that clear to everyone? So if we were to find a new species and we're wondering which domain it belongs to, all you need to do is sequence the DNA. And then that sequence will tell you which domain it goes into, because it'll be much more genetically similar to the other members of its domain than to any member outside of its domain. All right, we're going to talk about classification into one other category, and it's lower than a domain, and it's a kingdom. Now, there are different kingdoms, different number of kingdoms you can use. It depends on which your author is. I'm going to use the six kingdom approach, largely because it's simple, and to make the higher order kingdoms, all you need to do is separate the protista into more kingdoms than just the kingdom protista. But there are different number of kingdoms. There's a three kingdom approach, which means one domain is one kingdom. So bacteria is both a domain and a kingdom. Archaea is both a domain and a kingdom. Eukarya is also both a domain and a kingdom. There's just not much point in having a three kingdom approach because we already have the three domain approach. If you break the domains into the six kingdom approach, the domain bacteria has the kingdom bacteria, meaning the domain and the kingdom are the same. And that's true for the domain archaea. It will have the kingdom archaea, and the kingdom and the domain are the same. But then you have the domain eukarya, and it has 4 different kingdoms. The kingdom plants, plantae, the kingdom animals, and Amelia, the fungus, kingdom fungi, and then the protus, kingdom protista. Now, with that stated, I should mention that a protist is defined as a eukaryote that is not a plant, not an animal, and not a fungi. So the kingdom protista, it was always known that this is not a monophyletic group. And you can split the protista into different groups, and the higher order kingdoms do that, like the 15 and the 20. All of the other kingdoms come from splitting the protista. Now, the higher order kingdoms, like 30 or more, do split more than just the protista. The 30 or more split the kingdom plants into at least 2 different kingdoms, and it splits the archaea into at least 3 different kingdoms, and it splits the domain bacteria into many different kingdoms. Any question about any of that? All right, your author says that the different domains have different cell types. The domain bacteria has a cell type prokaryote, meaning that these cells are prokaryotic cells. The archaea also has the kingdom prokaryotes, meaning the archaea, their cells are prokaryotic cells. And then there's the eukarya, and it has eukaryotic cells. Now, your textbook author calls this a cell type. However, some authors call it a super kingdom. And you could divide all living things into a super kingdom, prokaryote or eukaryote, and then you can separate the super kingdom into the domains, and there will be 3 domains. And then further, you could separate the domains into different kingdoms. Any question about any of that? I usually call the prokaryotic cells and the eukaryotic cells a super kingdom, because that term was in use way before people started sequencing DNA, and then people proposed the 3 domain system. So the super kingdom was in existence long before the domains were known. Let's talk about some of the characteristics of the domain bacteria. All of the domain bacteria cells are prokaryotic cells. They have peptidoglycan in their cell wall. So if the bacteria has a cell wall, you can find peptidoglycan in their cell wall. The bacteria mostly multiply by binary fission. That's where one cell literally splits into 2 to make 2 daughter cells out of 1 parent cell. You can use the domain bacteria for making organic chemicals. Let me back up a minute. The bacteria get their energy needs from all of the possible energy sources. They can get their energy needs from an organic chemical, such as glucose, and we get our energy needs largely from glucose. They can get their energy needs from an inorganic chemical like nitrate or nitrite or sulfur, or iron. And some bacteria can get their energy needs by photosynthesis. Most of the domain bacteria are free living decomposers or primary producers. But some bacteria are parasitic and some are pathogenic, meaning that they live in and on us and they may cause disease. Any question about any of that? If not, let's move on to the characteristics of the archaea. They are prokaryotic cells. We do not find peptidoglycan in their cell walls. Peptidoglycan is only found in bacteria or domain bacteria. Some of the archaea are found in extreme environments, like the methanogens. They're found in methane environments. The extreme halophiles found in extremely salt concentrations. The body of water which has the highest salt concentration is the Dead Sea. The Dead Sea is found between Jordan and the West Bank. Some of the archaea are extreme thermophiles, meaning you can find them in extremely hot places and they survive and grow in that area. But when we looked further, we initially found the archaea only in extreme environments. But when we look further, we found that all environments on the planet Earth have archaea. And they are a significant portion of the plankton and the soil bacteria. Now, I said significant portion, they're not the majority of the prokaryotic cells. To my knowledge, they're not the majority anywhere, but that could be wrong, and I simply have limited knowledge. Here's a question for you. Are any of the archaea pathogenic? Do any of the archaea cause human disease? Nobody knows. Nobody wants to take a guess. That's either a yes or a no answer. I'm surprised nobody wants to take a guess.
I would guess yes.
Do you think some of the archaea are pathogenic? Why don't you guess again?
No.
No.
No is correct. The archaea, there are no known members which cause human disease. So none of the archaea are pathogenic. Because
Noted.
of that, I'm sorry?
I said noted.
Okay. Well, all right. The archaea do not cause human disease, and because of that, after this lesson, we will barely ever talk about the archaea, because the primary focus of this class is to look at microbes that cause disease, and the archaea does not cause disease. All right, let's talk about some of the characteristics of the eukarya. Remember, we're going to divide the eukarya into 4 different kingdoms, all under the domain eukarya. You can divide it into the kingdom plants, the kingdom animals, the kingdom fungi, and the kingdom protista. All members of the eukarya, all cells lack peptidoglycan. You only find peptidoglycan in the domain bacteria, in their cell walls. Let's talk a little bit about the kingdom fungi. The fungi use organic chemicals for obtaining their energy, largely glucose as the organic chemical. And that should be familiar to you because we obtain our energy needs the same way. The fungi include the multicellular molds and the multicellular mushrooms. Mushrooms actually do consist of masses of mycelia, which are composed of filaments called hyphae. And all of these cells make up the mushroom. The fungi also include the unicellular yeast, shown here. Most of the fungi are decomposers, but some of them, few of them, are pathogens, meaning a few can cause human disease. The only one I'm going to mention that at least I recall of a fungi, which we'll talk about in this class, is Candida albicans, which normally can be found on your skin as part of your normal microbiota, but you can get a fungal disease like a skin rash, or it can be under the nails or on the tongue or in the eyes. The point is that some of the fungi are pathogens and some of them can cause fungal disease. Candida albicans is the only one I think we're going to mention in this class, and it can cause yeast infections, especially of the skin, but of other parts of the body as well. Let's talk about the kingdom Protista. We can separate the protists into three different types. The plant-like protists, and those are the algae. The protozoa, or the animal-like protists, and then the fungus-like protists. The algae protists all engage in photosynthesis. They have cellulose in their cell walls. They all engage in photosynthesis. I think I already said that. And they do produce oxygen and organic compounds like glucose. The protozoa, on the other hand, the animal-like protists, many of them can move, like amoeba, paramecium, euglena. Amoeba moves by amoeboid motion, or what do you call that? Drawing a blank here on what that's called. When the cytoplasm is pulled in on one end and then protruded on this end by a pseudopod. So the amoeba can move by pseudopods. The paramecium can move by cilia. The euglena can move by flagella. Now realize that the eukaryotes can move in three different ways. I should say eukaryotic cells can move in three different ways. If the bacteria move, the only way that it can move is by flagella. The protozoa, as already stated, absorb and ingest organic chemicals like glucose, and that's what we do. The protozoa may be motile, and many of the protozoa are pathogens, where they cause disease. The fungus-like protists I'm not going to talk about other than giving you an example. And potato blight is a fungus-like protist. Potato blight, if you've never heard of it, it's what caused the potato famine in Ireland, which because the people were starving because the potato blight hit their potatoes, so they didn't have enough potatoes to eat. And so large numbers of Irish came over to America to escape Ireland and starvation. Any question about any of that? All right, let's move on to the domain eukarya. We can separate the domain eukarya into 4 different kingdoms. The first kingdom I want to talk about is kingdom plants. You can separate all plants into mosses, ferns, conifers, and flowering plants. All of the kingdom plants are multicellular, which I didn't mention it, but we did mention that some of the bacteria can be colonial. I gave you the example of Volvox. We do call the colonial bacteria and archaebacteria. Even if it's colonial, we call it unicellular, because if you break off one cell, that cell can reform the colony. And that's why we call it unicellular, because you only need one cell for life to remain, and the one cell will simply regrow the colony. All plants are multicellular, the same with all animals, meaning all animals and plants are composed of more than one cell. And that's the case ever since the fertilized egg cell split into 2 cells. So all humans are multicellular, at least once the fertilized egg becomes a 2 size cell. The kingdom plants we're not going to talk about much in this class because they do not cause any human diseases. I'm talking about a disease, not maybe a stomach ache from indigestion or something like that. All right, let's move on to the kingdom animals. You can split the kingdom animals into sponges, the worms, also called helminths, the insects, and the vertebrates. Now, we will continue using that system, but please understand that the worms should actually be split into 3 different types of worms. You don't need to know the annelids because we're not going to talk about them earlier or later. Why did I say earlier? Obviously, we haven't talked about it earlier. But the worms can be split into the roundworms and the flatworms. These 2 worms are as genetically different from each other as they are genetically different from humans. So we're putting them together and calling them worms, but they should be separate because they are a genetically different group causing the disease caused by the worms. And we will talk about kingdom animal. Specifically, we're going to talk about the worms in more detail because these worms can cause human disease. All right, let's reiterate that what comes under the umbrella of a microorganism is the bacteria and archaea, of course, are microbes. Viruses and prions are also microbes, but please understand they are not organisms. Viruses and prions do not have cells. Certain of the eukarya are a microbe. All of the protozoa, the molds, and the yeast are single cell. Or you can take the mold cell and split one cell off, and it will regrow the mold colony. Some algae are microbes, microorganisms, and that's any that's small. But the large algae is not a microbe, like the kelp that blows up on the Washington beaches. That is not a microbe. And then the helminths are considered microorganisms, even though most of the adult worms are not microscopic. For example, the beef tapeworm, which causes a tapeworm in a patient, can grow up to 30 feet or 30 meters. I forgot my units there. I'll have to look that up. But it's at least 30 feet. So the helminths, even though the adult worms are usually not microscopic, we do consider them microbes because the infective stage of the worm is usually, if not always, the first, I sort of lost my train of thought here. The helminths, the stage which is infective, is a microscopic. So either a person consumes an egg, like a tapeworm egg, and then gets the infection of the worm, or the parasite is microscopic and it can burrow into a patient. The point is that the helminths, the stage which is infective in people, is microscopic. And then helminths also cause diseases, and microbiologists study disease-causing agents. And so they do study the helminths. All right, let's talk briefly about viruses. As I mentioned, viruses lack a cell, so they are acellular. They do have a genome, and that genome either consists of DNA or RNA, but it is never both DNA and RNA. That genome is surrounded by a protein coat. The coat may be surrounded by a lipid envelope, but you should realize that only some viruses have a lipid envelope. Many do not. And when do viruses replicate? They only replicate when they have infected their host cell. Viruses are not a part of the three-domain system of life because they're not a cellular-based life form. Let's talk a little bit more about the three domains. So here we're looking at domain bacteria, domain archaea, and domain eukaryotes. We can see that the domain bacteria has a cell type prokaryote, the same with domain archaea, but domain eukaryotes has the eukaryote cell type. When we look at the chromosome of bacteria, we usually find one circular chromosome. So it's a circle, and the beginning place is the same place you end. That's why we call it a circle. The bacteria have no histones on their chromosome. A histone is a protein found in eukaryotes, and the DNA actually wraps around the histone protein. The archaea on the other, well, let's talk about the archaea. Also have one circular chromosome, You don't really need to know this, but they do have a histone protein. The eukaryotes, on the other hand, does not have a circular chromosome. They have a linear chromosome like the chromosome X, and the beginning point of that X is different from its endpoint. And that's a linear chromosome. Of course, the eukaryotes have histones, and they also have many chromosomes, not just one, which most bacteria and most archaea only have one. If we look at the ribosomes, the bacteria and the archaea have a smaller ribosome of 70S, and the eukaryotes have a larger ribosome of 80S. The S stands for sedimentation coefficient and an ultracentrifuge gradient, which probably doesn't mean much more to you than the simply labeling it 70S and 80S. The cell wall, as I mentioned, in bacteria has peptidoglycan. Peptidoglycan is not found anywhere in the archaea or the eubacteria. So as long as this peptidoglycan is only found in the bacteria. All right, let's briefly talk about the history of microbiology. The history of microbiology largely concerns the debate whether microbes could spontaneously generate or they had to come from a pre-existing life form. We'll also talk about the golden age of microbiology, which is early in the history of microbiology. You don't need to know the dates anytime except for the first date. And the first date you should know, the first record of a microbe, and actually the first record of life on the planet Earth, comes from fossils where we can find in rock the first fossils of the ancestors of bacteria, which is actually shown here, and that's an artist's rendition, or shown here. And that happened a little over 3.5 billion years ago. So you do need to know 3.5 billion years ago is when we have the first record of life on Earth, and it was the ancestor of a bacteria. The next record we have of a microbe is records from Babylonian clay tablets where they wrote out recipes for making beer. And you don't need to know this date, but that happened in 4300 BC. The Babylonians didn't realize that they had microbes which fermented the beer, but that's how they made the beer. And the first record is the Babylonian clay tablets. And then the next record is the Mayans made a fermented beverage from cacao. And to get cacao fermented, they were using microbes. Of course, they didn't know they were using microbes, but that's how they fermented cacao. If you don't know, cacao is what we use to make chocolate. And the cacao has to be fermented to get the chocolate. All right, any questions about anything? If not, I'm going to stop this lesson here. And we'll pick up here next time in the lecture. Any questions? If not, I'll say goodbye. Some of you I'll see in the lab at 7 o'clock.
Thank you.
Bye.
Thank you.
Bye.